I begin by thanking the organizers, Debashish, Prabal and Ambarish, for organizing this very nice event, particularly after 2 difficult years. It feels great to be physically present at a place like this and taking part in this conference. The title of my talk is Signaling Noise in Bacterial Chemotaxis. I will explain what is signaling noise, what is bacterial chemotaxis, everything in detail. Most of the results are done in collaboration with Shobhandev Mondal, who is a grad student at SN Bose Center in Kolkata. What is chemotaxis? Chemotaxis is motile response to chemical gradient. So there are some species, particularly we will talk about E. coli bacteria here, that which show this motile response in response to chemical gradient. So consider this picture where you have a buffer solution. In it you have some bacterial cells. Now if you dip a capillary filled with attractant in this solution, then the bacteria tend to swarm around the capillary. Similarly, if you dip a capillary filled with repellent, then they would like to go away from the capillary. This motion of E. coli happens using run and tumble motion. So what is that? A typical size of an E. coli cell is 2 micron, and there are some 10 to 12 flagella which are associated with each cell. Each flagella, they are attached to the cell at their base via a tiny motor. This is called a flagellar motor. When the motors, all of these motors, when they rotate in the counterclockwise direction, then they form a nice bundle like shown here, and this propels the cell forward. This is called a run. During a run, the cell swims smoothly in the medium. On the other hand, when some of the motors rotate in the clockwise direction, then there is no nice bundle, the bundle gets dispersed as shown here. This is called a tumble. During a tumble, the cell just undergoes random rotation about its body axis without much net displacement. So this is how a typical trajectory of a E. coli cell looks like. Suppose it starts from here. It is running now, the flagella are all bunched together. After running for a while, it tumbles here, then chooses a new direction to run, again tumbles here, chooses another direction to run, tumbles here, keeps on running like this. When you place such a cell in a gradient of an attractant or a repellent, as the case may be, then runs are elongated in the favorable direction and shortened in the opposite direction. And this is how the cell manages to show a net chemotactic drift. So this has been illustrated in this picture. Here in the left panel, we see an E. coli cell in a homogeneous environment. And you see it does a lot of run tumble, but after a large number of runs and tumble sessions, it comes back to roughly where it started. On the other hand, here by using this color code, we have shown a gradient, chemical gradient, and it is an attractant gradient. So whenever you are running downward, those runs are elongated. So after few runs and tumbles, the cell actually has moved in the direction of increasing attractant level. So this is the intracellular signaling network which makes it possible. I will explain this network. It has mainly 2 components, 2 modules. One is called sensory module and the other is adaptation module. The job of the sensory module is to, as the name suggests, it senses the signal and responds to it by modulating its run-tumble motility. On the other hand, the adaptation module makes sure that the levels, various protein levels, etc. inside the cell do not get too high or too low. It stays at an average value, and I will explain that it does so using the mechanism of negative feedback. So suppose in the extracellular medium, I have written stimuli here. There is it can be some nutrient molecule, some attractant molecule, etc. Suppose these molecules are there. They bind to the transmembrane chemoreceptors. And after binding to this ligand molecule, the receptor changes its conformation. We will describe the receptor in 2 states, active and inactive. And this nomenclature has been made based on their ability to help in phosphorylation. When the receptors are bound to stimulus, they are in the inactive state. They cannot aid to phosphorylation. On the other hand, when there are no stimulus present or very low concentration of stimulus present, then the receptors are in the active state, and this key A is a cytoplasmic protein which undergoes phosphorylation as a result of this activity of the receptor, and they become key AP. This key AP can donate its phosphate group to two different proteins, key B and key Y. One is part of the adaptation module, another is the part of the sensory module. Key Y, in its phosphorylated form, key YP, binds to the flagellar motors and increases their tumbling bias. The cell tumbles. So therefore, whenever the stimulus concentration is low or there is not much stimulus, the cell tumbles. In the opposite limit, when it is bound, then key AP cannot get formed, key YP cannot get formed, there is tumbling bias is low and the cell tends to run smoothly. Let's come back to the adaptation module now. Key B takes the phosphate group from key AP and becomes key BP. This then demethylates the receptor. It reduces its methylation level, and key R, another enzyme which actually methylates the receptor. And the negative feedback comes from the fact that only active receptors get demethylated and inactive receptors get methylated. So a little more quantity, yes? So yes, if it is repellent, then in the direction in which repellent concentration is increasing, those runs are shortened and the opposite runs are elongated, such that I can get away from it. Sorry, I always initiate with a tumbling motion. Yeah, tumbling actually allows me to reorient my direction. So if it is a good run, I keep going. If it is a bad run, I tumble and choose a new direction. Tumbling is just you are around this point and doing this. No, actually it can be a jittery movement also, yes, but there is not much movement, so to speak. You are just at a yeah, when it is repulsive, then yeah, everything is just opposite to what I said. When a repellent binds to the receptor, it increases its activity, which in turn increases the tumbling bias. It will be like that. Yeah. So this is a little more quantitative description of the previous network. So as it is clear to you by now, the two modules, sensing and adaptation, they are coupled via the receptor activity. And active and inactive states of the receptor, people have been able to write down the free energy difference between the two states. And this is a function of the ligand concentration and methylation. So this is the form of the free energy. And these are all constants, which can be experimentally determined. Let me just say what this means. So Kmin and Kmax, these constants, they set the range of sensitivity. If the ligand concentration is much larger than Kmax or much smaller than Kmin, then the cell cannot sense those range of concentration. And the switching rate between the two states, active and inactive, that depends on the free energy that separates the two states, such that in the adapted state, when the cell is well adapted to its environment, the average activity, which is defined as the probability to find the receptor in the active state, is given by this. This comes from simple Boltzmann distribution. So 1 plus e to the f inverse average of that. And these things I have already mentioned, that the methylation dynamics involves binding and unbinding of key R and key BP enzymes to the receptors. And active receptors get methylated. So imagine activity was high. Now active receptors get methylated. So M is going down. There is a negative sign here which is increasing this F, which in turn decreases the activity. So this is the negative feedback that I was talking about. And another thing to keep in mind is large activity increases the tumbling bias. One important aspect of this network is cooperativity shown by the chemoreceptors. So these receptors which are present inside the cell, they are not present as monomers, they form dimers. Now 3 receptor dimers, they come together to form trimer of dimers. And few of such trimer of dimers, they form a signaling team, or we call them receptor cluster. And all the receptors which are part of one cluster, they switch their activity together. So the free energy for a whole cluster is just the sum of free energy of all the constituent receptor TDs, trimer of dimers. And this is written like this. Here n is the number of such trimer of dimers present, 3n is the number of dimers, and the free energy is the sum of free energy of all the TDs. And inactive to active transition rate has been chosen from local detailed balance, and this is proportional to 1 by 1 plus e to the f, and the reverse transition, active to inactive transition, is e to the f by 1 plus e to the f. And this makes sure that the average activity has this Boltzmann form which I had shown in this slide. Now, one important reason for this cooperativity, this cluster formation, is that it amplifies the input signal. Instead of one receptor changing its activity, a whole bunch of receptor changes its activity, which has a direct impact on the tumbling bias of the cell. So this is actually, people have seen it, and this is actually understood to be the reason of very sensitive response shown by E. coli even when a weak concentration gradient is present in the medium, because it amplifies the input signal. So this methylation dynamics, I have already mentioned few things. One thing to keep, another thing is that each receptor dimer, they had its methylation cannot increase infinitely or decrease infinitely. It always has to be an integer and between 0 and 8. So therefore, if a receptor dimer has already methylation level 8, then even if a KR binds to it, it does nothing. It is already maximally methylated, and then the response also tends to get saturated. However, there is one problem here. The number of KR molecules is very low, only 140. Number of KB molecules is also around 240. Compared to this, there are few thousand receptor dimers in a cell. Then the question comes how the adaptation is maintained, because it seems like each receptor dimer has to wait for a very long time to get any attention from an enzyme, which are few present in such a less number. So to explain this, these concepts like assistance neighborhood and brachiation had been proposed theoretically, and also some of these things have also been observed experimentally. Assistance neighborhood, now the problem is if I do it like this, each enzyme molecule comes, binds to a dimer, changes its methylation level, and again comes back to cell cytoplasm. Again another binding event takes place. If it goes on like this, then clearly each dimer has to wait for a very long time to bind to an enzyme, which is going to affect the adaptation properties of the cell. So assistance neighborhood suggests that whenever an enzyme is bound to a receptor dimer, it can also modify the methylation level of all the dimers which are present in its neighborhood. And brachiation suggests that a bound enzyme can perform some kind of a random walk on the receptor array. And along its trajectory, whenever it is landing on one receptor, it modifies its methylation level. It does that for a while before unbinding and coming back to the cell cytoplasm. So these two mechanisms have that explain why E. coli cell manages to maintain such robust adaptation. Finally, the run and tumble motility. I have already said that the QIP molecule, which actually is responsible for the tumbling bias, this is the time evolution equation for QIP concentration, but actually these reactions are happening pretty fast. So you can as well think that for each activity, dyp dt is 0, quasi-equilibrium, such that yp has this expression. So which means whenever activity increases, tumbling bias increases. So the picture that I want you to keep in mind is high activity means large tumbling bias, short runs, and low activity means long runs. Now the question that people have been asking for some time now is what is the effect of noise on single cell chemotaxis? So the first question that I have to answer is, what is the most important source of noise? Now for a long time people believed that the most important source of noise is methylation. Why? Because these methylation demethylation reactions are very slow. They are 100 times slower compared to any other reaction step in the signaling network. So therefore any fluctuation which is happening at that stage comes as a slow noise, and it is very difficult to get rid of a slow noise. And fluctuations are also something I would expect here, because I just said that the enzyme molecules are present in really low concentration. So therefore they are subject to fluctuation, and those fluctuations come as a slow noise. I cannot get rid of them. And in fact, in an earlier work, we had considered the role of methylation noise on chemotactic performance. However, about 5 years back, these two groups, they performed independently, they performed experiments where they had shown even when there is no methylation, they are still recording significant activity fluctuation. They used a freight-based setup to directly measure the receptor activity, and they had shown that even when you switch off methylation, demethylation part of the network, there is still significant fluctuation. However, when you disrupt the receptor cooperativity, when you do not allow them to form this cluster, the activity fluctuations are negligible. So therefore, this experiment concludes that receptor clustering is an independent and equally important noise source in the pathway. And the question we are asking is how this newly found noise source affects different aspects of chemotaxis. So in this talk, I will mainly focus on these three questions. How this newly found noise source affects chemotactic efficiency. I will describe in a moment how do I quantify efficiency. Secondly, I will ask how does it affect the methylation or activity dynamics. Now, one thing is, although E. coli chemotaxis people have been looking at for a long time, we have found that there is no systematic study of quantifying or correlating the activity fluctuation or methylation fluctuation inside the cell to what is happening in its ligand environment. So we would like to do that, and we would like to ask how this dynamics is affected by this newly found noise source, namely receptor clustering. These two questions are mainly relevant for swimming cell. Third question is for a tethered cell, and here we ask how does a tethered cell is affected by this receptor clustering, particularly how does it short time response to a step stimulus is affected. So I will just elaborate on each of these things. First, I will summarize my results, main results, and then I will show you the data, the details. First question was effect on chemotactic performance. How do I quantify a single cell chemotactic performance? I ask how fast the cell is able to climb up the gradient. That is the chemotactic drift velocity. Or I can ask how strongly it can localize in the nutrient-rich region. Or in general, I can measure the ability of the cell to distinguish between the regions of high and low attractant levels. So you can see all these things are like performance criteria, and we find that there is an optimum size of the receptor cluster at which each of these performance criteria show a peak. Okay? And we explain this observation from a competition between the two modules, sensing and adaptation modules in the signaling network. We show that these two modules compete with each other, and as a result of this interplay, the system shows a performance peak. So this work was published last year in this journal. Second question that I am interested in is how this sensing adaptation interplay affects the methylation dynamics. I wanted to probe the interplay further. How does it affect the methylation dynamics? And we measured the temporal variation of methylation level as the cell navigates through different chemical environments. And particularly, we correlate the methylation or activity dynamics to the changing ligand environment, which has not been done before. And we find that there is a very sensitive dependence on receptor clustering. In particular, when the ligand gradient present in the medium is strong, the sensing adaptation competition gives rise to highly non-trivial methylation dynamics. And these results were published this year, at the beginning of this year actually, in this journal. And the third question we ask is, what is a short time response to a step stimulus? So, most of the experiments on E. coli happens on tethered cell, okay? Although people do look at swimming cell experimentally too, but most of the experiments happen on tethered cell. So here also we consider a tethered cell and we just give it some stimulus, and the quantities which experimentalists measure typically are two quantities, receptor activity and clockwise bias of the flagellar motors. And immediately after giving the stimulus, these quantities change rapidly, reach some extremal level, and then in the long time limit slowly adapt to the post-stimulus adapted state. Most of the experimental and theoretical work actually focus on the long time relaxation. We focus on the short time behavior. Particularly, we are interested in the extremal condition, which is achieved shortly after the stimulus has been administered. And in this limit, we have been able to perform some exact calculation, and we find very good agreement with our numerical simulations. And we find, one of the interesting results we find in this case is that there is an optimum size of the receptor cluster at which this extremal response is reached in the shortest possible time. So these results are presently under review. So now I come to my data. So first, we wanted to see that as in our model, as I increase the receptor clustering, we indeed have higher activity fluctuations, what they had seen in experiment. So I just plot probability distribution of receptor activity as a function of, for few different values of the cluster size. And as you can see from this data that indeed when I have larger and larger clusters, the distribution is wider. In the inset, I plot the variance of the distribution as a function of the cluster size and find an almost linear dependence. Now these results are not really surprising, because as you have larger clusters, this also means you have fewer number of clusters, because total number of receptors in the cell is fixed. So therefore, the total activity that you are calculating by summing over activity of all the signaling teams, now you are summing them over smaller number of signaling teams. So the fact that variance grows with n linearly is nothing but central limit theorem. So although in those experimental papers it has not been mentioned, but I think it is pretty easy to see why one would expect more noise when the receptor clusters are larger. So here I have plotted the localization and drift velocity. So I consider a linearly varying attractant concentration profile as shown here. Now if the gradient is not too strong, then I can expect the probability distribution of the cell position, Px, is also linear. In fact, in this inset, I show Px, it is indeed roughly linear with x, and the slope of this distribution is actually a measure of chemotactic performance. If the slope is large, that means the performance is strong. So I plot the slope here, and as you can see, at a specific value of n, at a specific cluster size, this slope shows a peak and then it comes down. In the lower panel, I have plotted chemotactic drift velocity. Now, E. coli has a run and tumble trajectory. So there are different ways you can extract drift velocity from there. You can measure the net displacement in a run and divide it by the average run duration. That could be one measure of drift velocity. Or you can just observe the cell for a fixed time interval t, large enough, and measure the net displacement of the cell in the time interval and divide by t. So for both the measures, I find there is an optimum value of n at which drift velocity is highest. From there, it is coming down. There are a few more performance criteria that I have measured here, and all of them, these things are actually measuring the differential behavior of the cell when the nutrient level in their environment goes up or down. I don't want to get into too much of details, but the net result is all these things show a peak at a specific n. Now this is the explanation. The free energy that I have written down, if you remember it, it had two parts, one coming from ligand, another coming from methylation. So I write it in this way, Fl minus Fm. Now, as the cell swims uphill or downhill, by uphill or downhill I mean as it is moving towards higher concentration level or lower concentration level, the change in FL is proportional to N, because if you remember the expression that I had used, let me just quickly go back to that, here. You see there was an N here because I was summing over all the free energy of all the dimers. So therefore this change in ligand free energy actually it is proportional to N, which means as N increases, the change in free energy is larger, the ligand part. So the activity now changes more quickly, and therefore favorable runs are elongated even more, bad runs are shortened even more. This improves the performance. But large N also increases activity fluctuations, and at some point adaptation has to now become very active I mean alert, because now activities are fluctuating wildly. So therefore now this methylation part has to play a more dominant role, and often this methylation is actually controlling the variation of free energy rather than FL. So now the cell is less sensitive to what its ligand part is telling it to do. It is more sensitive to what its adaptation part is telling it to do. It cares less about whether ligand concentration is increasing or decreasing. So this is the data to support this argument. Here I have plotted change in free energy, both FL and FM separately, as a function of t, as a function of time for first few steps of an uphill run. In fact, I have looked at first 0.5 seconds of an uphill run. The continuous line shows the change in FL, and these discrete points shows the change in FM. And as you see from here, when the cluster size is small, the methylation change is much less compared to the FL change. It starts catching up as you increase the cluster size, and finally for large clusters, it's really much large. So in this limit, the cell hardly cares about what is happening to FL. It is completely dominated by the adaptation module, and its performance goes down. So what we have seen, enhanced sensitivity, which is a result of clustering, comes at the cost of increased biochemical noise. And this gives rise to an optimality, optimal strength of cooperative interaction. In some previous work, people had actually reported an optimal cooperative interaction for best chemotactic performance some years back. But they had considered a noisy input signal. And their argument was that because of receptor cooperativity, the input signal is being amplified. But if there is noise in it, noise is also getting amplified. So too much of amplification is not good. And therefore, beyond some point, cooperativity does not help me. But we are finding an optimality whose origin is completely different. Notice that here in our model, we did not consider a noisy ligand environment. Even then, we are finding an optimality which is coming from the interplay between the two modules of the signaling network. Next, we want to ask how the sensing adaptation interplay affects the methylation dynamics. So let me first carefully define the exact quantity that we had looked at. So here, MIT is the methylation level of a particular receptor cluster measured at time t during the ith uphill run. So t is equal to 0 means when the run is starting, and I keep track of the methylation level of any cluster, and this plus and minus superscripts, they are denoting whether the run is uphill or downhill. And n plus minus t is the number of runs which are persisting till time t. So this capital delta mt, it measures it considers the run, let's say an uphill run, it measures the methylation level at time t. From that, it subtracts the initial methylation level of the same run at time 0, and it does average over many such runs, sum over I, and finally it divides by the total number of runs taken. Compared to that, small delta m is this quantity. And you see here what it does, it looks at the methylation level at time t, but from it, it subtracts the average methylation level of all the runs. So the main difference between these two quantities is that at what am I subtracting? At t is equal to 0, I can either subtract the initial methylation level of all runs or initial methylation level of all persistent runs. And as I will show that this is a crucial difference which can completely change the dynamics. So this is my data. I will not describe in great detail everything that I have here, but this is the capital delta M data, the first quantity. The left panel is for uphill run, right panel is for the downhill runs for different values of n. So one common feature about which is present in all these panels is that first there is a slight tendency, first it becomes negative slightly, and then for large t it becomes positive. So first there is a small tendency of demethylation, and then the methylation takes over. In fact, this is not so difficult to understand because at short times there are many runs. There are many short runs which are present in my population, and those short runs, why they are short? Because they tend to tumble quickly, and those things tend to tumble quickly which have high activity, and high activity means demethylation. So therefore, at small time, overall there is a tendency of demethylation. But as time becomes moderate or large, then I have these long runs whose activity should be low, and therefore they should be more prone to methylation, and as a result I am finding this kind of behavior. When I plot small delta m, the other quantity, I see something very surprising. You look at this first panel, n is equal to 10. Here it was initial demethylation followed by methylation. But here it is demethylation all along, it's just dropping. So actually what is happening here, although individual trajectories, they may initially get demethylated, then they go on to methylation, but also systematically many runs are terminating and dropping out of the population. And that is particularly those runs which have high activity or high methylation level. So this is a scenario where you can compare it to some social situation where you have a group of people, and suppose everybody is saving money, but the richest people in the population keep dying at a given rate. So therefore the population average of the wealth continues to go down with time, although everybody is saving money. So this is something like that. Although we see that tendency gets different for larger n, but that is because that individual growth is so strong in that case that even though the top methylation trajectories are getting out of the population, it still preserves the trend. Okay, let me not go into that. This is the data for strong gradient. I will not explain everything here because I don't have enough time for that. I just wanted to show this to, convince you that how non-trivial the methylation dynamics can be. So particularly you see, if you look at the uphill runs, it is still qualitatively similar from what you had seen earlier for the big gradient case. But the downhill runs, depending on the cluster size, are completely different. They don't even look, they look nothing similar to each other. In fact, look at this one, n is equal to 100. This is a perfect showcase of sensing adaptation interplay. What happens here is for short times, there is strong demethylation, but then that strong demethylation brings the activity so low that now methylation has to take over. So now it starts getting methylated again, this thing turns over, increases, but then at this, when the time is really large, like 2.5 seconds or 3 seconds, then a downhill run is happening in face of a strong gradient. So the drop in the ligand free energy along the trajectory is so much, now it can actually take on methylation. And under the effect of that, it is again dropping down. So maybe everything is not clear here, but I just wanted to point out, we understand all the features here, but I don't want to go into too much of details in this talk, but I just wanted to point out it is extremely important. Receptor clustering qualitatively change the methylation dynamics. These things I don't need to get into. Then this brings me to the last point that I wanted to study, is the short time extremal response. So this is the response to step stimulus at short times. So what is the kind of situation I have in mind? I have a cell which I have tethered at a point, and then the cell cannot move. So there is no gradient here, okay? Because the cell cannot move, no point in having spatial gradient of attractant. But around the cell, I suddenly change the attractant level. I can increase it or decrease it at time t is equal to 0, and I hold it at that level for t larger than 0. Now in the pre-stimulus state, when t is less than 0, or in the long time limit, t goes to infinity, the cell is adapted to its environment. And then the receptor activity and clockwise bias, the two quantities which are of interest to experimentalists, they have time independent average values for t less than 0 and t goes to infinity. But at short times, right after I have given the stimulus, these quantities show rapid variation, they reach extremal values. And I am interested in the question, how long does the cell need to reach its extremal response, and how to characterize the properties of the signaling network at that time. And we have been able to do exact calculation for this limit, and it agrees very well with our numerical data. So this is our data. Here I have plotted, the left panel is for step addition, right panel is for step removal. So left panel is where I have increased the attractant level from 200 to 250 micromolar, and right panel is I have decreased it from 300 to 200 micromolar. I have plotted the receptor activity as a function of time. As expected, in the pre-stimulus state, activity was not changing with time. I have plotted two things here. The discrete symbols are for receptor activity and the continuous lines are for this function. So you see, if the free energy is now also a function of time, because I have, first of all, because of changing the ligand concentration, I have given it a sudden change, and after that adaptation happens because methylation now tries to catch up. So free energy is also a function of time now. So I also measure this quantity, and these continuous lines are data for that. And these three curves are for three different cluster sizes, 5, 10 and 15. So as expected, and this was known actually, people had seen it a lot of times in experiment, there is a rapid variation, extremal value followed by a comparatively slower relaxation. And as expected in the pre-stimulus state, both these things, activity and this free energy expression, both of them were equal because the system was adapted. As soon as I apply the stimulus, they start differing, but at the extremal point, they again become equal. Now this is slightly strange because I expect these two quantities to become equal when the system is adapted. But short time after the stimulus, when it is extremal deviation I am observing, when actually I can rephrase it by saying that it is farthest from any adapted state. Even then I find these two quantities become momentarily equal before they start getting away from each other again. So this equality is expected in an adapted state, and the fact that I find it here, this time, I call it actually TA, the extremal time. So this is the exact calculation to explain this phenomena. Let's say Pn1t is the probability to find N1 active receptor clusters at time t. This is the time evolution equation for Pn1t. This is the master equation. And here, gamma 0 to 1t is the total transition rate from inactive to active states. Remember here, I have to consider transition rates which are happening for all possible values of methylation. So Pmt given 0 is the conditional probability that given a cluster is inactive, what is the probability to find its methylation level m at time t. And then Wa by 1 plus e to the f is actually the inactive to active transition rate for that cluster, and finally I sum over all m. Similarly, I can define gamma 1 to 0 also. So this is the time evolution equation for this quantity, and then I multiply with n1 on both sides and sum over all n1. So you can see that, it may not be obvious from here, but it is actually pretty easy to show that will cancel lot of terms. In physics, we call it telescopic cancellation. Lot of terms get cancelled, and finally I have this very much simpler equation. So here ddt of n1t average, that is the average number of active clusters at time t. However, when I divide it by the total number of clusters n, that is nothing but average activity. And at time t is equal to Ta, that quantity is hitting its extremal value. So this time derivative has to vanish at time t is equal to Ta. So using that and just 2 steps of simple algebra actually gets me to this result, that average activity calculated at t is equal to Ta is equal to this free energy expression calculated at the same time. So an equilibrium-like relationship between time varying activity and free energy holds momentarily when the system is farthest away from any adapted state. Next, I focus on Ta, the extremal time. And here I plot Ta as a function of n, the cluster size, both for step stimulus and step removal. Step addition and step removal, sorry. And in both the cases, I find there is a minimum. So, which means there is an optimum size of the receptor cluster size at which the extremal response is reached in the quickest possible time. And next, I want to explain this effect. So first, let me define this quantity Zt, which is the difference between this Boltzmann form and average activity. By definition, Zta is 0 because I have just proven that at t is equal to Ta, these 2 quantities are equal. So first I will explain why for large n, Ta increases with n. So here is it. Consider the case of step removal. You can easily generalize it to step addition also. For step removal, when you have large n, then the drop in F is so large because you have suddenly changed the attractant level. Drop in F is so large that this F becomes a large negative number. Then e to the F you can almost neglect. So therefore, this quantity becomes very close to unity. This means that active to inactive transitions are effectively blocked. When this is almost as good as unity, active to inactive transitions are effectively blocked. So Zt, which was defined like this, it now becomes 1 minus average activity. And in this time range, At can only increase during this time. And as n increases, as you have larger, and so the drop in F is also larger, and therefore it takes longer for the system to lower its methylation level such that the reverse transition is possible again. So I explicitly show this here, where I have plotted this quantity, this quantity explicitly for as a function of time for different n. And as you see, when n is large, it remains stuck to unity for a very long time before it actually comes down. So therefore, the time during which this reverse transition remains blocked increases with n, and this increases Ta. For large n, Ta increases with n. Next, I will argue for short n, Ta decreases with n. So here, when n is small, then no such thing happens. When n is small, this quantity remains significantly below unity, no transition gets blocked. Both forward and reverse transitions are possible now, but the rates are function of n, and the difference between these rates actually increase with n. So activity now increases faster, and the variable Zt that I defined, that reaches 0 quicker. So therefore Ta now decreases with n. So Ta decreases with n for small n, increases with n for large n, so there has to be some minimum in between. However, there is an interesting outcome of this argument. Everything that I have said so far can be repeated if I just hold n fixed and play with the step size of the stimulus. I can say that for a fixed n, if I have a large step, then there will be larger drop in free energy and one transition will remain blocked, everything goes through. So which means if I fix n and vary the step size of the stimulus, there is an optimum size of the step stimulus at which the extremal response is reached in the shortest possible time. And indeed, in our simulation, we verify this. This is for a fixed n, we just play with change the step size, and you see around 40 micromolar, there is a minimum. And this thing can be easily tested in experiments. It may be slightly more complicated, although not impossible, to vary the cooperative interaction, but you can easily change the step size of the stimulus that you are giving, and then measure how long does it take to reach the optimal value. So this is something which can be easily tested in experiment. So these are the results for the other quantity, clockwise bias, which also experimentalists measure. So here we have plotted the time evolution of clockwise bias. It also remains constant in the pre-stimulus state, decreases sharply, reaches a minimum, both for step addition and step removal. And we call this extremal value at that time, we call it TCW. We can exactly calculate this extremal condition also. I will not go through all the details, just tell you the outline of the calculation. Here QCWN1T is the joint probability to find the motors in the clockwise rotation mode and with N1 active receptor clusters at time t. You can write down the time evolution equation for this joint probability, and it will have two kinds of terms. One is because of transition between clockwise to counterclockwise mode, and another is the transition for the active inactive state, which changes the value of N1. However, when you sum over all N1, these active, all these terms which are containing these N1 transitions, they cancel. And you finally have a simpler expression. Here this QCWT is the exact quantity that we measure, probability to find the cell in the tumbling mode at time t. And this quantity has this simple form, and when at t is equal to TCW, when it is hitting the extremal point, the time derivatives should vanish. So these two things should be equal. So we independently measured these quantities in simulation and we plot it as a function of time. And indeed we see the time at which it crosses 0 is same as the time TCW that we had measured in this type of measurement. Okay. Even in this case, TCW shows a minimum with n, although here error bars are larger and as a result the minimum is slightly shallow. Significantly shallower compared to what we had seen for the receptor activity case, but you can still see that there is a minimum. So finally, to conclude, I think I finished before time. Most experimental and theoretical studies, they consider long-time behavior of the post-stimulus recovery. We have shown that short-time response is important and it provides useful insights into the adaptation kinetics. And we believe that using tethered assay and Fred-based setup, our prediction of optimum step size can be verified. And in fact, these experimental groups, they have also been able to tune the cooperative interaction between the receptor, the strength of the interaction. So we believe that it should also be possible to actually check that indeed whether there is an optimum strength of cooperative interaction at which this performance peaks, or at which the extremal response is reached in the shortest possible time. And another thing is, so far for E. coli, no in vivo measurement has been possible for methylation level. And from our result that extremal activity actually equals to this free energy, the Boltzmann form of free energy. Using that result, one can actually get some kind of an estimate for what is the free energy value when the activity is at its extremal point. And then one can actually estimate the typical methylation level for this case. Okay, and yeah, another thing is, we have not used too much of modeling details in our calculation. So the calculations do not, for example, take into account all these binding, unbinding kinetics of enzymes to the dimers, which we have actually used in our model, but we didn't. So therefore, we believe that the calculations that we have are quite general, and even for some other kind of models, they should remain valid. This is the acknowledgement for funding from DST. So with that, I would like to thank you for your attention and wait for questions.
We have a lot of time for questions. You were saying that this long time duration, so how much long
Long time duration means? Oh, no, no, actually, yeah, what I meant is that, you see, it is a sharp drop, then it slowly increases, okay? So most of the experimental and theoretical studies, they focus on this long time relaxation, when it is close to, how does it relax? That's what they are focusing on.
It is in minutes like that.
It is in, it can, yeah, either those, that is, yeah, it can be in minutes, actually. I think so. Because, for example, people look at half maximal, like, I mean, what is the target value that you want to reach, and how long do you take to reach 50% of that? But even that happen, and then people analyze those time scales. Those are actually pretty well characterized. But what I meant is that this time, what happens immediately after you apply, not too many, I mean, there are actually not too many studies on this. Particularly, yeah, these extremal conditions, etc. These things, how long does it take to reach this extremal point? These things people have not looked at. Yes.
Maybe I missed it, but you talked about clusters of these receptors?
Yes.
That flip together.
Yes.
But you didn't say how they form, like how they, is there like an Ising model that explains how they form?
Yeah, that's a good question actually. In fact, in our model, we start with these clusters.
And you fix them all at the same size?
Fix the value, yes. But we have checked that even if you use some distribution, but they do not change dynamically. I mean, you can draw the sizes from a distribution, our conclusions do not change much. This we had checked. This data that I had shown here is for fixed cluster size and all same value. But what we plan to do next, that it's like what you said, Ising model, that actually dynamically they can form and build. Yeah, that we plan to do next. Yes. So I think for E. coli it is half an hour. In a very good condition, it is 20 minutes, and generally it is considered to be 30 minutes. I don't know. I don't know. All the, sorry?
All the chromatin marks are being removed, and then the DNA is being.
Yeah, could be. I don't know enough about this actually. I definitely I have not considered this here. Different relaxation, sorry. This one? Yeah.
So are there experiments where people see different kind of like this relaxation time scale in experiments?
Yeah, sometimes people consider different experimental conditions. For example, I know that they change the size of the step stimulus and they see the relaxation. I don't think by changing cluster size it has been done, or for different cooperativity it has been done. I have not seen that. No, for yeah, it should be possible, because now people can actually tune they can, I mean, for example, in this experiment, that the data which actually discovered this new source of noise. So what they have done here is they have disrupted the clustering, and they have actually measured the activity fluctuation. So in this experiment, actually they, I mean, using this type of setup, actually it should be possible to tune the cooperativity.